So I'd like to continue with the work of our collaborative group at Utah. Um, and let me start out by saying that the work we're doing is really based on the hypothesis uh, of Rob Fair and Ron Davis uh, as to what's going on in ME/CFS. And I've been on the advisory board of the Open Medicine Foundation for a long time now. And I was always looking for an opportunity uh, to potentially use our collaborative group for a drug development or discovery project. Uh, and the problem was that we really didn't understand the disease very well. And so it was essentially impossible to carry out a drug discovery project without some additional knowledge about the mechanism of the disease. And then Rob came up with the itaconate shunt hypothesis. Uh, and we really got very excited about that because for the first time, there seemed to be an opportunity to carry out a drug discovery type project that had the potential to benefit patients. So let me just tell you in a few minutes why we were so excited about the Rob Fair hypothesis, because it really seemed to us to explain a lot of the very mysterious features of ME/CFS. So the itaconate hypothesis is that, of course, the citric acid cycle is the source of energy generation in all cells. And the citric acid cycle or Krebs cycle really kind of behaves like a gasoline engine. You eat your food, that's reduced carbon. You fill an automobile gas tank with gasoline, that's reduced carbon. And basically, the citric acid cycle does the same thing as a gasoline engine. It essentially oxidizes the reduced carbon completely to carbon dioxide, and that releases a lot of energy. And of course, that's what allows your automobile to move. The Krebs cycle also, of course, releases a lot of energy, and that energy, as Rob explained, is stored in the form of ATP. And the level of ATP in all cells, the level of production, determines what that cell can do. And so if you have a muscle cell and you want to move, the amount you can move is really dependent on how much ATP you can produce because movement consumes ATP. So the explanation for fatigue that the itaconate shunt provided really was very attractive because it said that the reason you feel fatigue when you're sick is because this itaconate shunt is turned on. And when it's turned on, the Krebs cycle is unable to produce quite as much ATP. And as a result, your muscles don't produce as much ATP, and therefore this is the direct cause of fatigue. And so it's sort of exciting to us that there was a normal mechanism for inducing fatigue when necessary. And it made a lot of physiological sense that if you were sick, that there should be a mechanism to encourage you not to run a marathon because you needed all your ATP for fighting an infection that was dangerous. And so the specific form of the hypothesis that Rob first came up with was that an infection viral infection, bacterial infection, would result in the expression by cells that were infected of interferon alpha, which is a signal that says we have a crisis here. And that then caused the production, induction of expression of ACOD1, which led to a decrease in ATP and therefore less muscle motion, a feeling of fatigue, and so on, and also the production of itaconate. So the detail, I think, of why what's going on in ME from the sort of 10,000 foot level is that normally a viral or bacterial infection turns the itaconate shunt on, and so you have lowered ATP in a subset of your cells, and so you feel fatigue. After infection, normally this shunt is turned off, and therefore ATP levels are restored, and your feelings of fatigue disappear. But in fact, what they hypothesized was the pathological response was really the failure to shut off the itaconate shunt. And so if after infection, the itaconate shunt fails to turn off, then ATP levels will remain very low, and therefore you'll have a chronically low level of ATP, and that would account for chronic fatigue. What was really attractive about this hypothesis was it also explained why there was so much variation in the clinical presentation of ME/CFS. Because if your infection, for example, were in the lungs, which, for example, is the case in long COVID, then your lung tissue would be the most drastically affected, and the very low levels of ATP, as they continue, would cause all kinds of pathologies. While if your infection were in your GI tract, then your GI cells would be most affected, have chronic low levels of ATP. And of course, the longer you have a shortage of ATP, then you have downstream symptoms that could account for a lot of the diverse pathology of ME/CFS. So if we're going to do any kind of drug discovery, the first order of business is to identify a molecular target. And of course, that job has been done for us by Rob and Ron, who then show that if we're able to target ACOD1 so that it's not active, then the itaconate shunt won't turn on, won't be effective, and therefore the levels of ATP in the cells that are affected will be restored to normal levels, and that presumably would be beneficial to people suffering from ME/CFS. So the first order of business, if you're going to do a drug discovery program, is to develop an assay for the bioactivity of the molecular target. And our group, collaborative group, has developed two assays, a microbial assay in the Kelly Hughes laboratory and a biochemical assay by the Eric Schmidt laboratory. And so the Kelly Hughes lab showed that if you expressed ACOD, here called human CAD, in E. coli or Salmonella, then you got a phenotype, a readout. So if you grew the cells in glucose, whether you induced the enzyme or not did not make such a big difference. So the gray curve here is when the enzyme is induced. But if you were growing the cells in acetate, then the ACOD expression made a very big difference. And you could see this difference in a plate. And so if you did not induce the enzyme, the colonies would grow and you'd get a little colony in a petri dish. But if you induce the enzyme and you grew the cells on acetate, the colonies would not grow. And so what that meant is we had an assay, because if you inhibit the enzyme, then presumably now you would get growth. And so this is an assay that we can use. It's a plate assay to very efficiently screen a large number of compounds as to whether or not they affect ACOD1. The Schmidt laboratory took a more rigorous biochemical approach. They purified the enzyme, and then they were able to come up with an analytical method where they could see both the substrate and the product. And so the enzyme converts cis aconate to itaconate. And so here's the substrate coming off here, and here's the product. And so you can follow as a function of time what's going on. And so here's the substrate, and it's converted to the product. And so you can follow the biochemical assay in a very quantitative way. And so we now have two assays, and now we can begin to try to identify compounds that alter the bioactivity of the molecular target, which is ACOD1, to characterize the interaction between each compound and the molecular target, namely ACOD1. And for chronic fatigue syndrome, because there is no drug that's widely effective, we decided to take a variation of the usual drug development strategy. And so what we did was, instead of looking at all compounds, we preferentially looked at compounds that would not require the full drug development route. So normally, of course, the route for drug development requires 3 phases of human clinical trials, and that takes typically a few years. But what we decided to do was first look at drugs that were already approved. And because they're already approved, any clinician can use this drug for a new purpose. And what we found was, when we screened a library of FDA-approved drugs, we could identify one FDA-approved drug, which in fact affected ACOD1, inhibited ACOD1. We also decided to look at natural compounds, herbal preparations, supplements, etc., and that's still ongoing. But we have one candidate identified, a natural compound, and presumably this will, because it's a natural preparation, it will not require the 3 phases of human clinical trials. So we prioritize that, but in parallel, of course, these are not necessarily the best drugs. We've also done the conventional route and looked at libraries of other compounds that I'll tell you about. So that's the overall workflow of what we've been trying. And so we started screening a 2,000 member library of already approved drugs. And we used the microbial assay developed in the Hughes laboratory. They identified 22 initial hits using that assay. And then the Schmidt laboratory looked at these 22 hits with a more rigorous biochemical assay, and they found 5. And then looking in more detail at the dose response, we have one FDA-approved drug which seems to inhibit ACOD1. And so that's where we are with FDA-approved drugs, and the compound, I'll call it compound 1, is an FDA-approved drug that inhibits ACOD1. We also identified the natural metabolite which inhibits ACOD1, and I'm going to call that compound 2. Now, once you've identified compounds that look promising, these have to be tested. And importantly, you want to test them in vivo in animal models. And so we had to develop 2 animal models. And Randy Peterson has told you about zebrafish. And Chino Raguraman has developed a mouse model that we can use to test the most promising compounds. And so this kind of in vivo work, after you've identified the compound that affects the enzyme, which is a kind of a test tube test, you really need to test it on animal models of the disease. And you've heard, of course, the advantages of using zebrafish. But let me use a few minutes to do a mouse model, to describe the mouse model. So it's based on the fact that interferon alpha induces ACOD1. And so what Chino did was he injected mice with interferon alpha, and then every day he injected another dose. And now he asked the question, does this affect the behavior of the mice? And the assay that he used is the running wheel assay, where you put mice with a running wheel and they run, and you can record how much they run. And so this is quite quantitative. And what you find if you observe the mice is that as they become more familiar with the running wheel, they tend to run more. And so this is the kind of thing you see at day 1. This is how much they run. And as they get used to the running wheel, they increase the amount they run, and then that plateaus off after about 4 days. In the animals that were injected with interferon alpha, you can see that there's a significant decrease in how much they run. And so this is essentially the assay that we're going to try to use. And so what I'm going to try to show you is the effects of adding compound 1. And so here we've treated the mice with compound 1 in addition to interferon alpha. So here are the untreated controls. Here are the injected animals, and here are the animals that were treated with compound 1. And what you can see is that compound 1 results in a reversal of this deficit in movement. And at the end of 5 days, these are moving just as much as the control animals. Now, we also injected itaconate, and as Rob has described, itaconate probably contributes to the pathology of ME/CFS, because when we inject the mice with itaconate, they don't move at all, in fact, at this dose. And furthermore, even though we inject compound 1, we can't reverse the lack of motion. So what that tells us is that compound 1 is really an inhibitor of ACOD1. If you inject the toxin produced by ACOD1, and Rob's hypothesis is that this is itaconate or itaconyl-CoA, then you're not able to reverse that with compound 1. So compound 1 acts before itaconate is produced. on the ACOD1 enzyme. So that's our conclusion. And so what we now know, I'm going to skip these slides, which document why we think itaconate is demonstrating that itaconate is toxic. But we now think that it's not only the lowering of ATP, but the production of itaconate that leads to the pathology of chronic fatigue syndrome. We've done biochemical characterization of compound 1. We know that it competes for binding with the substrate, and therefore, in the presence of compound 1, the enzyme is inhibited. We have a few problems that we're still trying to solve. So this is compound 2 that shows that as you increase the concentration, you inhibit the enzyme. And this is compound 1 in blue. And what's we still don't understand is the shape of this curve, because it suggests that there's a more complex mechanism of inhibition. And so we're still trying to understand that. So in summary, we really have two drug leads. One, an FDA-approved drug, which presumably, if it looks good enough and passes a few more tests, clinicians can begin to try it on their patients. And we have compound 2, which is a natural metabolite and which will require a little more in the way of FDA approval. This, if we feel that it's safe for clinical use, can be directly used by a licensed physician. So let me tell you where else we're going. We're looking at herbal extracts of various types. We're beginning to screen 100 extracts that are used for Ayurvedic medicine. And so we should get additional hits from that. And in addition, the Schmidt Laboratory, for example, has huge libraries of potentially bioactive compounds. They have a library of what are called circular peptides. And so these are new compounds, but these libraries are vast. One library can have as many as 2 million compounds. And we are producing mixtures of these compounds and are testing using both the microbial screen and the biochemical screen, trying to identify active components of these compounds. So once we identify an active component, because these are peptides, we can very rapidly improve the properties, the specificity, and the potency of the compound. And so this is a normal drug development pathway in which eventually we should be able to get an ideal drug. But this pathway of development, of course, takes the usual clinical trials that the FDA mandates. So as you can see, we're trying a really broad program. We're committed to generating new compounds that hopefully will reach the clinic. And by prioritizing FDA-approved drugs, we believe that there will be something that can be tried on patients and hopefully is effective on a wide variety of patients, because the Fair-Davis hypothesis suggests that ACOD1 is central to the pathology, and therefore, if we can inhibit it, we're hoping that a lot of patients will be benefited, and that not only will we reverse the ATP deficiency and the production of itaconate, but the pathologies that result from prolonged ATP deficiency, it's hard to predict how those will be reversed, but we're hopeful that some of those pathologies will be reversed as well. So to summarize, we now have two compounds that look promising. We're validating those with in vivo assays, like the two animal models, and we need to work out dosing, toxicity, safety, etc. And then we're looking at libraries of so-called alternative medicines, and then conventional drug discovery moving forward. So that's an overall picture of the program of our collaborative group. I think you can see that it's a broad program. We're very optimistic that that it will be productive, and that hopefully we can identify compounds that will be used clinically for therapeutic interventions, hopefully as soon as possible. So with that, the main PIs who are involved in our collaboration are listed here, and here a lot of people in our lab who've done some of the work. And again, we would really like to thank Khosla Ventures for supporting the work that we have done so far. So thank you very much for your attention, and I hope that I've conveyed what we feel is the present state of our drug discovery and development efforts. Thank you.